Time for lab number 3 for our AP2. And today we're doing a couple different blood tests. We're going to start out by talking about blood typing and doing some blood typing experiments. So, what kind of formed elements do we see here? You should definitely know these. These are red blood cells. We can see their biconcavity that they've got going on here. Definitely red blood cells. Super abundant cell in our blood, super abundant cell in our body. Now, all cells, red blood cells included, are going to have antigens on their surface. So all of our cells, all of our red blood cells are going to have antigens on their surface, so surface antigens. So a surface antigen can be of many functions and uses, but one helpful thing to consider is a surface antigen can serve as a little flag on the cell. A little flag letting the immune system know that it belongs. So every red blood cell will have a variety of little surface antigens on it. They'll be of different shapes, different sizes, all sorts of different things. Now, for our purposes today, we're not worried about, a whole ton of surface antigens. We're worried about 3 major red blood cell antigens. There are 3 major ones. There are lots of minor ones, but only 3 major ones. And these red blood cell antigens, why they're important is they are going to determine your blood type. Like, for example, my blood type is type O positive. And the letter here, the letter that I've got, is determined by the presence or absence of these 2 antigens, antigen A and antigen B. So whether you have the A, the B, the both, or neither is going to determine the letter of your blood type. Now, the third one is called antigen D. It's also called antigen RH. It determines the symbol of the blood type, whether your blood type is a plus or a minus. So you've either got just A and B, just B, or neither of these 2. And that particular combo determines the letter. Now, I'm type O. So what do you think about me? Do I have the A? Do I have the B? Do I have both of them on my red blood cells, or do I have neither of them? Think about that for a second. Come up with a guess. We'll answer it in a second. Now, with the antigen D, you either got it or you don't got it. And that determines the symbol. Now, I have a plus as a symbol. So do you think I have this antigen, or do you think I do not have it? Again, make a guess, and we'll find out. All right. So here are 8 possible blood types based on these 3 antigens. So notice that all of my guys on the bottom here, all 4 of my red blood cells on the bottom have the RH, also known as the D antigen. Remember, RH and D are the same thing. So all my guys on the bottom have the RH. So all my guys on the bottom get a plus sign. So if you have the RH antigen, the D antigen, on your red blood cells, your blood type gets a plus. So I told you a moment ago that I was positive. So that means my red blood cells have this RH antigen on them. Now all my guys up top, none of these 4 have the D. No D, no RH on any of these 4. So notice how all of these guys get a minus. So that's the symbol. Symbol's pretty straightforward. If you got the D, you get a plus. If you don't got the D, you get a minus. Okay, now let's deal with the letter of the blood type. This guy has neither A nor B. Same down here. My 2 guys right here in this column have neither A nor B. This guy has A but not B, B but not A, both A and B. So if you have neither A nor B, like these two gentlemen here, you get O as your letter. If you have neither A nor the B antigen, okay? So type O is going to have neither A nor the B antigen. So if you are type O, you've got neither the A antigen nor the B antigen. So I'm type O. I'm type O positive. So because I'm type O, I don't have the A or the B. Now I'm positive, so I got the RH. If you're type A, like the second column here, if you're either A minus or A positive, you have antigen A. Whoops. You've got antigen A, but not B. If you are in this column, you're type B, you've got antigen B, but not, oh, that's terrible, not A. And if you have both A and B, then you're type AB. So, whether I have A, B, A and B, or neither determines the letter of my blood type, and we have 4 possibilities here. O if you got neither, A if you got the A antigen but not the B, B if you got the B antigen but not the A, and AB if you got both of them. And then the symbol is determined by whether you've got the D antigen. If you got it, you get a plus. If you don't got it, you get a minus. All right, so notice how AB positive has the most antigens. It's got all 3. O negative has got the least antigens. It's got none of these major ones. Of course, don't forget, these are just the major antigens. This red blood cell, it's not like it's empty on the outside. There are lots and lots of minor antigens, but we don't really worry about them. All right, good stuff, good stuff. Here is just another representation. If I'm type A, I've got my A antigen. If I'm type B, I've got my B antigen. Notice that B has no A's and A has no B's. If I'm type AB, I've got both of them. And if I'm type O, I've got neither one. Now, what we now have to bring into the story are antibodies. Now, you may or may not have talked about antibodies a little bit already in lecture. Antibodies are immune proteins that mark antigens for destruction. So antibodies are these immune proteins. They grab onto antigens. In fact, they're going to grab onto foreign antigens, right? You wouldn't want to grab onto your own personal antigens, would you? Because then you would destroy your own cells and tissues. Now, just like there were 3 major important red blood cell antigens, for today's purposes, there's 3 major antibodies. There are anti-A's, anti-B's, and anti-D's. Remember, anti means against. So anti-A antibodies will bind antigen A, marking the red blood cell with it for destruction. Anti-B antibodies are going to bind antigen B and mark any cell with it for destruction. And anti-D antibodies are going to bind antigen D, marking any cell with it for destruction. So a good rule of thumb is that you have antibodies that target the antigens you lack. So you have antibodies that target the antigens that you lack. Okay? All right. So let's consider me, for example. Me, what's my blood type? My blood type is O positive. So Does my blood's plasma have anti-D antibodies? Do you expect me to have anti-D antibodies in my plasma? Cross my fingers here for your answer. No. I'm not going to have any anti-Ds. Because think about it. What does that plus sign mean? That plus sign means that I've got the D antigen. So I cannot have the antibody that would attack it, right? But I'm type O. So I lack the A and the B antigen. So I'm going to have anti-A's. I'm going to have anti-A's. And I'm going to have anti-B's. Okay? Because that's what I'm missing. So like, if you were type A and you gave me your blood, I mean, thanks for the offer, right? But it's going to turn out bad. If you're type A and you give me your blood has A antigens. That's foreign to me. My immune system is going to attack it. My antibodies, I've got anti-A's that are going to attack it. All right, definitely think about this. Absolutely think about this. So if we check out in my graph here, if I'm type A, I've got the A antigen, and I've got the anti-B antibody. If I'm type B, I've got the B antigen and the anti-A antibody. If I'm type AB, I don't have either one of these antibodies. And if I'm type O, hey, that's me. If I'm type O, I've got both of them. Now, in a moment, we're going to do something. For now, I want you to notice that these antibodies, imagine that each of these little curvy parts here, kind of like half circle parts, they're like the grabby parts, okay? So this one antibody can grab onto 5 different antigens. Keep that in your brain for a second, okay? And here we see some red blood cells mixed with some antibodies. And let's just pretend, you see these little dots here? Let's pretend that little dot is antigen A, okay? That little dot is antigen A. And this antibody, let us pretend it is the anti-A antibody. So what's going to happen is the two tops of the Y here are going to grab on to the antigen. And this is going to do 2 things. Like I said earlier, it tags these cells for destruction, but it also creates what's called an agglutination. The fact that these antibodies can grab onto more than one cell creates a clump of cells. And a clump of cells is more likely to get destroyed by macrophages. So that's called agglutination. And agglutination is a sign that antibodies have grabbed onto red blood cells, that there was some sort of mismatch going on. All right. Good. One thing we got to consider is that my anti-A and anti-B antibodies, and anybody's anti-A's and anti-B's. So if you're type A, you've got anti-B's. If you're type B, you've got anti-A's. If you're type O, you got both these guys. Amongst antibodies, these are weird. They are weird antibodies. They're weird antibodies, okay? They're weird because they are said to be preformed, which means they're made without exposure. Usually your immune system makes antibodies upon exposure. So like, you get a particular strain of the flu virus that you've never seen before. Hey, you're gonna get a little sick, but your body's gonna make antibodies, it's gonna do other things in response to that exposure. So most antibodies are made due to exposure. These guys are weird in that they're not. The word for that is preformed. Okay. Now, in terms of blood transfusions, like giving blood, donating blood from one person to the next, we got to make sure the recipient's antibodies do not attack the donor's antigens. So we got to make sure the recipient's antibodies don't attack the donor's antigens. Okay? So we're concerned with the antibodies of the recipient and the antigens of the donor. Okay, time out for a second. Who would be the best donor? Who would be what we call the universal donor? Well, think about it for a second. Come up with an answer. Don't look it up. Just think about it. Use your brain. And if we're concerned about donors' antigens, well, who has the least antigens? Who's got neither A nor B? Type O. And also does not have RH. Type O negative is the best donor because someone with type O negative, they've got none of the three major antigens. All right, who is the universal recipient? Okay, think about it again. We're concerned with what? We're concerned with the recipient's antibodies, right? Donors' antigens, recipients' antibodies. Well, who's got the fewest antibodies? Well, that would be the guy with all the antigens. Someone who is AB positive. All right, good stuff. So considering blood donations, this is a handy picture. What it's telling me is that everybody can donate to themselves. Like O can give to O, A can give to A, B can give to B, AB can also give to AB. You can always donate to yourself. And because O is a universal donor, notice how O can give to everybody. So O can go to everybody. And because AB is the universal recipient, AB can receive from everybody. So this is a helpful visual way to know which blood transfusions are safe. You should write this out, draw it out, because you're going to be asked in your exam about blood transfusions. All right, as far as the RH goes, well, we got two situations, RH negative and RH positive. Of course, if you're negative, you can give the negative. Of course, if you're plus, you can give the plus. And the only other thing that can happen is minus can go to plus. In fact, there's only one thing that can't happen is plus cannot go to minus. That's the only thing that cannot happen. All right. So in lab, we're going to run an experiment where we're going to have some unknown blood. Like this is an unknown blood here, okay? We're going to figure out its blood type. To figure it out, we've got bottles of antibodies. This blue one has got anti-A antibodies. Remember, anti-A's grab onto and agglutinate the A antigen. We got anti-B antibodies, which grab onto and agglutinate the B antigen. And we got our anti-RHs, which grab on to and agglutinate the RH antigen. And we're going to put some blood, like blood number 1, we'll put some blood here, here, and here. Then we'll add a different antibody to each of these little wells, and we'll look and see if agglutination happened. If agglutination happened, like so, for example, let's say I put this blood in here, and I put anti-A's. If I see agglutination, it means the anti-A's had something to grab onto, which means the blood must have had the A antigen. And depending on the combination of agglutination that I see, it's going to let me determine the blood type. All right, let's walk through some possible situations. So what I did here is I put blood, a blood sample. It's the same blood sample. I put the same blood sample in all 3 of these wells. Okay, I put the same blood sample in all 3 of these wells. And then what I did is I put anti-A antibodies, like In this guy, I put anti-B antibodies in this one, and I put anti-RH, anti-Ds, anti-RH in this one. Now my little red lines here indicate agglutination. So that means the anti-A's must have grabbed onto something, which means the blood must have provided the A antigen. So this blood is going to contain, this blood must contain the A antigen. If I see agglutination, I know that antigen is present. I see agglutination here. So the anti-B's must have had something to grab onto. The blood must have provided the B antigen. And lastly, I see agglutination here, which means my blood must have supplied RH antigens for the anti-RHs to grab onto. So my blood sample in this case had all three antigens. So my blood sample in this case is AB positive. All right, let's run through all the possibilities here. In this situation, I see agglutination in my A well and my B well, which means, the A and B antigens must have been present, but no agglutination RH, so this dude is AB negative. All right, let's do another one. What if I see agglutination in the A well and the RH well, but not in the B well? What do you think that's going to be? That's going to be A positive. And if I see it just in the A well, not in B, not in RH, that means I just have the A antigen, so A negative. And what about this one? This one would be B positive. And this one, B negative. And this one, well, it's got neither A nor B, so no agglutination there. So neither of these antigens, which means is going to be O. It's got agglutination here, so it's O positive. Whoops. And our final one, nothing anywhere. So this blood provided no antigens, which means it's O negative. All right, folks, that is it for blood typing. The second test we're going to do is hematocrit. And hematocrit testing, of course, we're not really going to do it. We're going to use a simulated situation. But you would take blood. You would centrifuge that blood. When you centrifuge blood, you get it separated. You get the liquid part, which is plasma, buffy coat part, which is my white blood cells and my platelets, then my red blood cells. Now, hematocrit is the percentage of whole blood occupied by red blood cells. Usually in the low 40s for ladies, usually in the upper 40s to 50s for boys. Okay? Now, how do we actually do it? Well, after we centrifuge the blood, we've got our blood sample. One thing we can do is we can take a measurement, right? We can say, okay, red blood cells divided by all the blood, and I can divide that and turn into a percent by multiplying by 100. So in this case, red blood cells were 18, whole blood was 37. I don't know why I didn't make that 36. It would have been so much easier. 18 over 37 times 100. I want to say that's going to be what, like 48%? Bust out my calculator here. I could have pretended like I was doing this math in my head, but I'm not. We get 48.6%. So that is our hematocrit. So all we do is we measure the red blood cells. We use a ruler to do that. Also measure the entire blood. Divide red blood cells by blood. Times it by 100. Yeah. And we could tell if this person was anemic or polycythemic if we did this. All right. So here we got two examples where something has gone wrong. This guy is going to have a low hematocrit. That's indicative of anemia. This guy is going to have a high hematocrit, and that means polycythemia. And if you're in my lecture, you know we talked about those things in lecture as well. Beautiful. We're almost done here, coming to a close. Oh, if you want to do it without math, there's a math-free way to do the hematocrit. It involves this card here. Notice the card has these gradations going from 0 to 100. Well, that's where our percentage is going to be somewhere between 0 and 100, right? So what we're going to do is we're going to take our tube, and here's just another example of a card. We're going to take our tube and we're going to align the tube such that the bottom of the red, so here, see how there's the red and there's the plasma here. We align the tube so that the bottom of the red is on the 0 line, the top of the yellow is on the 100 line, and we just read the red. This is at 39% here. There's no math involved. And we have two different shapes here and this diagonal line in case we have different sizes of blood samples. Like this is a smaller sample size. So we align the red on the 0 line, the yellow on the 100 line. We read it and we get a little under 30% or so. That's all you do. Bottom of the red, 0 line. Top of the yellow, 100 line. Move it to wherever you need to achieve that. Then just read the top of the red. And that, everybody, is it. We are done with lab number 3. Thanks for coming. See you in the next one. Bye-bye.